It's a great honour to introduce Linda Birnbaum to you all tonight. Linda's the current director of the National Institute of Environmental Health Sciences in the USA, and arguably she's the world's premier, the NIHS is the world's premier environmental health research organisation, and Linda is also director of the National Toxicology Programme. So we're delighted to have you here, Linda, at Brunel. Just to say a little bit about Linda before she gets up and talks. What she said to me in the car was, just say, I'm a wife, a mother of 3 children and 3 grandchildren. So I will say that as well. But I just want to say a little bit about Linda's career. So she started it off doing a BA in biology at the University of Rochester in New York, and then studied microbiology for her master's at the University of Illinois. She continued on in Illinois to do a PhD, and attracted to the fields which were then burgeoning of molecular biology and molecular genetics. She mapped the ribosomal RNA genes of E. coli, and then she went to do a postdoc at the University of Massachusetts in Amherst, and was an assistant professor of science at Kirkland College in Clinton, New York. She then spent 4 years doing research into aging at the Masonic Medical Research Laboratory, also in New York, and it was here that she began her career-defining work in toxicology, in studying how altered metabolism could affect aging. She modulated the metabolism in rats using dioxins and polychlorinated biphenyls, PCBs. She looked at how these chemicals break down in the human body, and for the next 10 years, she explored the molecular effects of dioxins, analysing their relative toxicity of related chemicals and designing long-term bioassays to assess cancer risk. Linda moved to the National Toxicology Programme in 1979, and in 1989, after 9 years there, she became director of the Experimental Toxicology Division at the EPA, Environmental Protection Agency, and she held that post for 19 years. Taking over as the head of EPA's Environmental Toxicology Division, I'm told she expanded its ranks to an all-time high of 90 full-time employees and dozens of students and postdocs. Despite budget cuts, she found ways to continue toxicology studies by partnering with other federal agencies or academia. She spearheaded some of the first work documenting the mechanism of action of chemicals which disrupt hormonal systems, endocrine disrupting chemicals, such as brominated flame retardants. And she is the recipient of several Science and Technological Achievement Awards from the EPA. She began serving as the director of the National Toxicology Programme and concurrently as the National Institute of Environmental Health Sciences director on March the 13th, 2009. She was the first woman and the first toxicologist to hold this position of the NIEHS director. The mission, for those of you who don't know, the NIEHS is to reduce the burden of human illness and disability by understanding how the environment influences the development and progression of human disease. NIEHS focuses on basic science, disease-orientated research, global environmental health, clinical research, and multidisciplinary training for researchers. One of the many things I admire about Linda is that she is still an active and respected member of the scientific community. I also admire that she did all of this being a mother as well, and now a grandmother. She's had many positions in academia. She was vice president of the International Union of Toxicology, and I think she's now president-elect. Is that correct? That's what I read. She is the author of more than 700 peer-reviewed publications, book chapters, abstracts, and reports. And in addition to her role at NIEHS, she is also an adjunct professor in the Gillings School of Global Public Health and the Department of Environmental Sciences and Engineering at the University of North Carolina at Chapel Hill, as well as in the Integrated Toxicology Programme at Duke University. In October 2010, she was elected to the Institute of Medicine of the National Academies. This is one of the highest honours in the fields of medicine and health, as well as to the Collegium Ramazzini. She has numerous other awards, not all of which I'm going to list, for leadership in environmental health and policy, and also for diversity leadership, and she's also a real champion for women in science and won a Women in Toxicology Mentoring Award from the Society of Toxicology. Talking to Linda in the car coming over from the airport today, she told me that she is someone who believes in the synergy that can come from different kinds of science. So it's fitting that she is talking to us here at Brunel as we move closer towards the restructuring of Brunel to form the 3 colleges and the 3 interdisciplinary research institutes. We're going to have to do much better integrative science if we're going to address the challenges we're facing now, and that's what we were discussing on the way over, in amongst all of the climate change related storms. So without further ado, I'm going to hand you over to Linda, who's going to talk to you about environmental exposures acting like uncontrolled medicine. Thanks, Linda.
So what I really want to do today is just give you a brief journey through some of the issues of when environmental exposures can act like uncontrolled medicines. And I'm happy to answer questions either during the talk, well, probably better if we hold them to the end, about some of the issues that I'm going to raise. And I can only obviously touch on some of the issues. Whoops, well, we can go beyond that. So I think we know that modern medicine is incredibly good at treating disease and improving the quality of life. We have all kinds of drugs that we can use. We've got statins to lower cholesterol, and we've got antibiotics to treat certain diseases, and we've got something like tamoxifen, which can be used for receptor-positive breast cancer very effectively. But then on the other hand, our environment is also incredibly good at causing disease and impacting the quality of life. And those same systems that drugs act upon can also be acted upon by things in our environment. So using tamoxifen as an example, which is a strategic estrogen receptor modulator, we've got lots of environmental chemicals like BPA that are also SERMs and can do the same kinds of things as drugs that we use, and I think we have to remember that. One of the really important things that we have to understand is that there's a shift in what the environmental burden of disease is. For many, many years, much of the focus when we talked about environmental exposures is we were focusing on infectious agents. And this now is some data from the World Health Organization, and what you can see, let's see, I think this is, whoops, okay, that was not the pointer. This is the pointer. Okay, what you can see is that we have a tremendous increase in the burden of non-communicable chronic diseases. You know, in much of the world, certainly in the developing world, we've conquered or largely conquered all those infectious diseases that used to kill people by the age of 40. Remember, in 1900, the average life expectancy was 40. I just heard today, speaking to one of you, that in the UK now they're estimating that a child born today will live to the age of 100. But we know that in 2000, the average life expectancy was in the neighborhood of 70 to 80 for people, as compared to 40, and that's because people weren't dying early of many infectious diseases. Now, we know that in the less developed world, infectious disease can still be a major problem, but it is being conquered there. And what's interesting is that the chronic non-communicable diseases, things like obesity, type 2 diabetes, cancer, and heart disease, are now the major cause of disease and death throughout the world, and the role of that it's actually more important, for example, and the role of the environment, which WHO estimates is at least 30% determining the causes of disease, are actually greatest in some of the less developed parts of the world, so that there are bigger environmental impacts. And I can give clear examples of some of that. Some of the things in the developing world, or in the developed world like the US, we're not as concerned about acute high dose, you know, poisonings. But if you go to some parts of, for example, Africa, like in the Niger Delta, where there's actually oil just bubbling out of the grounds in places, and in those villages, there are no old people anymore because of the toxicity there. Or if you go to parts, for example, actually of Brazil and the Amazon, where they're doing gold mining and people are all dying from mercury toxicity. I mean, these are kinds of things that are still very important. So let's see, this was forward. So what are the health conditions that have the largest environmental burdens? And I don't expect you to read this, but the green line is with the environmental fraction. And we know that things, for example, and some of these are infectious, but some of them are not. And I think some of the point is we've got to start talking about the interactions, how an environmental exposure may make you much more susceptible to an infectious agent, or how an infectious agent may change your susceptibility. So for example, respiratory tract infections. 4 million people die a year, mainly children, from indoor air pollution, and largely that is driven by indoor cook stoves. And when I say cook stoves, that's kind of a euphemism sometimes in the less developing world for just, for example, a couple of stones that form a circle and people are burning dung or they're burning wood. For example, in the stoves, there's no venting other than maybe a hole in the roof. And 3.5 million children a year die from respiratory tract infections associated with this air pollution. And we know from animal studies, and you're going to hear me a number of times try to relate that we can learn from what we learn from our animal and our mechanistic studies informs our understanding of what we find in people. What we know is that exposure to wood smoke lowers the sensitivity of animals, to, for example, to bacterial infections, respiratory infections, or to influenza, for example, much more susceptible. So, and if you look at some of these others, for example, COPD, of course, the biggest contributor to COPD is cigarette smoking, but air pollution, I mean, for example, in Beijing, in Delhi, in other places with horrendous air pollution, we have lots of people who have COPD who've never smoked at all in their lives. For example, ischemic heart disease, clear evidence for environmental contributions there from not only from air pollution, but other things. But we could go through this whole age, and even if you talk about lung cancer, the estimates are now, forget smoking-related lung cancer, that air pollution, the International Agency for Research on Cancer a month ago actually concluded that air pollution is a known human carcinogen. So what do I mean when I say the environment? We tend to think, when people say environmental contamination, they tend to think we're only talking about chemicals. Oh, I have to mention, because I see my husband who's in the audience smiling, that's our youngest granddaughter. But, you know, it's not just industrial chemicals, it's not just agricultural chemicals, it's also, for example, prescription drugs. There's this dichotomy in people's mind. What are drugs? Drugs are chemicals. What are the nutrients in food that we eat? Made up of chemicals. They just have different uses and different utility. But lifestyle choices and substance abuse, obviously alcohol, smoking, today in the US where marijuana is becoming legalized in many states, these can all have impacts on our health. We know that I've already talked a little about air pollution and combustion. I mentioned infectious agents. What about the microbiome? This is a huge, hot, new area of research. You know, there are 10 times as more bacteria in our guts than there are human cells in your body. And what we're beginning to understand is that your diet has a tremendous impact on what your microbiome is. And we know that environmental chemicals, agricultural, industrial, et cetera, can impact your microbiome and alter that distribution. But we know that the microbiome actually handles many of those chemicals and may in fact make them worse or better for you. In fact, arsenic, which I'm going to come to later, which is a naturally occurring element, but is a carcinogen, reproductive toxicant, developmental toxicant, immunotoxicant, we could go on with the list, in fact is metabolized by bacteria in your gut to forms which are more problematic than possibly what you actually ingest. And then social and economic factors have to be considered as well. Whoops. So chemicals are widely dispersed in our environment, and I think one of the most important things is we are never exposed to one chemical at a time, and that's not only because one chemical, but we're all eating, we're all drinking, we're all being exposed to drugs and over-the-counter compounds and so on. And the important thing is a lot of the focus used to be on very high doses of exposure. A lot of environmental health initially came from occupational studies where our workers were being overexposed to many things. But what we're understanding now is the levels that are present in our bodies today, and depending upon which group does the biomonitoring, you can easily measure 200, 250, 300 different synthetic chemicals in our body at any one time. And I can tell you that with some of the exciting new techniques that are being used, in fact, you can identify over 20,000 different compounds that are present in 100 microliters of human serum. Tremendous number of things that we're going to understand. So I've already said that exposures don't occur alone. I talked about how infectious agents or the microbiome can alter our response to a given chemical. Well, one chemical can also alter our responses to another. Now, all of you probably have taken a drug at one time, and you read on the thing, if you're taking this drug, you better not have grapefruit juice, for example. Or it'll say, do not eat with meals, and that's because of the interactions that can go on between one chemical or one group of chemicals and another. And we've got to begin to look at the combinations, and the exposome is a new term that's being used to look at the totality of exposures for a person, and that's where we're getting this data on over 20,000 different chemicals that can be present in your serum at any one point in time. And I should mention that the UK is one of the leaders in exploring this whole new approach to understanding what do we really mean by human exposure. So we're also beginning to think about new ways of environmental health. We used to talk about the high dose exposures where you basically overwhelm the body's defenses by brute force. But now we're really beginning to focus on low doses of chemicals in where they can act like hormones especially, and can disrupt development and can disrupt the function of the basic physiological functions of bodies at low doses to which we're all exposed. Another point that we're beginning to focus on is that the exposures can occur at one point in your life, but you don't actually see the effects till much later in life. So the susceptibility to disease can persist long after the exposure ends. And this is a whole new science of epigenetics, which is you're not changing the basic sequence of the DNA, you're changing how the DNA is read out. It's changing in DNA expression. And we're beginning to understand, and this is really not Lamarckian, who was discredited, a century ago, but in fact, we're finding that these non-mutational events can actually persist from one generation to the next. We're also beginning to look at things more holistically. If you have a complex disease, a complex heart health condition, there are going to be multiple causes, multiple ways that you can achieve that condition, and that there are going to be multiple things going on. And we used to focus very much, certainly in the US from a regulatory point of view, the focus was all on cancer and birth defects. Nobody was much concerned about other impacts. But now we're beginning to understand that the environment can contribute to a whole host of different health endpoints, whether it's obesity or diabetes, and I'm going to show you some examples of this, cardiovascular disease, different impacts on the immune system and reproduction and neural effects, both neurodegenerative diseases as well as, for example, neurodevelopmental effects, as well as cancer and birth defects. So I'm going to talk a little and give you some examples about endocrine disrupting chemicals, which have been defined as an exogenous substance or mixture that alters the function of the endocrine system and consequently causes adverse health effects in an intact organism or its progeny, or possibly in a population. And a new report came out, and I think Susan and Andreas, were you both involved in this report from the World Health Organization just last year, which was a state of the science on what do we know about endocrine disrupting chemicals. And it was really a 10 or 11-year update on the first state of the science report that came out from WHO in 2002, because frankly, nobody even thought about this whole area of endocrine disruption until 12, 13, 14 years ago. But what WHO has concluded is that the endocrine disruptors are becoming a global threat that needs to be addressed, because our endocrine system helps maintain our basic functioning as an adult, but is absolutely critical during development. So sometimes when people think of the endocrine system, they tend to think about reproduction predominantly, but a point that I'd like to make is that it's very complex, and every endocrine system talks to every other endocrine system. You can't perturb one endocrine system without having some impact on another. There are It's very, very highly regulated at many points. It can be easily modulated, and these modulations can occur at very, very low levels of exposure. And what I mean by activational or organizational, I mean if the endocrine system gets perturbed during development, it's going to lead to a change which is not reversible. In an adult, for example, there's a lot of concern right now about men who've been taking testosterone supplements or using testosterone creams, but if you stop taking them, many of the effects will go away. But if you have altered levels of the male hormones during development of a male fetus, you're going to have adverse impacts that are not going to be reversible. I'm going to show you some examples of that. So there's some trends in human health that I think that we all need to be cognizant of. Richard Sharp actually pulled this together about 10 years ago now, but it hasn't changed. First of all, what we can see is that there is an increase in the incidence of testicular cancer and breast cancer from the early 70s, basically over a 20-year period. And there's also been an increase in hypospadias, which is a condition in where the tip of the urethra doesn't come out at the tip of the penis, but can come out somewhere along the penile shaft or even at the base of the penis. This is a birth defect. It can be usually quite well corrected, but it is a problem, and there is an increased risk with hypospadias. It's associated also with undescended testicles and potentially with testicular cancer. But the other thing is that there is a decrease, or there was a decrease in sperm counts that has been documented in many populations around the world. In some areas where the level has gotten really low, it may have leveled out, and actually we're dealing with issues of reduced fertility in men in some of these places. So what's happening also with reproduction? Well, guess what? I don't know how many of you have young children, young girls, or young grandchildren, but the age of menarche has decreased dramatically, clearly from the 1800s. Much of this in this range was related to improved nutrition, but then we've got almost a dramatic drop at the last half of the past century. And I can tell you a paper just came out from one of our grantees last month, Frank Biro, and following a cohort of 1,200 young girls, starting at the age of 6. The girls are now at the ages of about 14. And what they found, at least in the United States, is that African American girls are starting breast development, over a quarter of them by the age of 8, white girls and Hispanic girls by the age of 9. I don't know about you, but it's very hard, I think, to think that 8 and 9-year-old girls are ready to begin to mentally, they're not really ready to be starting puberty. But we're finding this is kind of occurring in much of the developing world, and obviously this has a lot of societal impacts as well. So there's been some studies looking at the impacts of environmental exposures on reproductive health, and this was again looking at the age of menarche here, and this was looking at, this was looking at PBDEs, which are a kind of flame retardant. The lower brominated flame retardants were used extensively and almost entirely in the United States and Canada. They were banned in the EU. I have to say, the EU is way ahead of the US when it comes to regulations of some of the environmental chemicals. But they were banned in the EU, and then the US, the sole manufacturer, decided to stop making them, mainly because there started being market pressure where people said, I don't want them anymore. But the major type of brominated flame retardant was something called DECA, which actually was also banned in the EU except for specific applications in about 2009 by an act of Parliament, the EU Parliament. But in the US, the producers voluntarily agreed last year to stop making them. But the point is here is this is based upon what the levels were in the mother during pregnancy, and what you can see is that as the levels basically increase from the lowest levels of exposure, and again, levels in the US of these are about 10 times higher than the levels of people in Europe, and it was a function of where it was used. And what you can see as the levels increase, what you see is that more and more of the girls are starting menarche at younger and younger ages. Now, we recently helped sponsor a cross-government effort which also involved scientists and physicians from outside the government and breast cancer advocates. You cannot do environmental kinds of work unless you get the advocacy and the community groups involved in your studies. And the result of this was to look at what kind of work is going on in the government, what is the funding related to breast cancer and the environment research, and what needs to be done. And I think the most important thing is the title of the report, which is Prioritizing Prevention. And that's something I'm going to come to again and again, because how much better to prevent, for example, breast cancer than having to treat it once it occurs. And so this looked at identifying science advances and developing a comprehensive strategy. And one of the key things is you've got to have multidisciplinary work going on here. You cannot do environmental health work without having people from multiple disciplines involved. And that, again, as I said, you need to get the public involved in these studies. So talking about breast cancer, what do we know about environmental exposures? And there's a lot of mechanistic data, there's a lot of animal data, and we're beginning to get data from human epidemiological studies indicating the role of the environment. And I think one of the strongest studies that not only pointed out an association with DDT exposure, which, as you know, is a pesticide that was banned in most of the world. It is still used a little bit in certain countries where they are having terrible times controlling malaria. But it's basically been banned. But everybody in this room has measurable levels of DDE, which is the metabolite of DDT in their bodies. And the point here is that if you look at when exposure occurred to DDE, to the DDT in these girls, there was no significant effect at all if it occurred once they were older than 14. The younger when the exposure occurred, the higher the odds ratio, the risk of getting. So for girls who were under the age of 4 when they had DDT exposure, and I'm talking about higher levels that occurred, we're talking about, this is data from a population of women who were recruited in the 1950s and the 1960s in the US. Serum and urine was taken from these women. They have come back repeatedly for follow-up studies. Their children and now their grandchildren are being studied. But the women who had higher exposure, say in the 40s and the 50s, had the highest risk of developing breast cancer, and it decreased as they got older. Now this brings up the issue of the importance of when exposure occurs. And we're beginning to understand that early life exposure is most likely to have the longest consequences. And I can tell you that every time I read an epidemiology study and they're measuring what is in the levels of chemicals in a person, say when they have cancer, I want to say, you know, that horse left the barn a long time ago, because cancer has, depending on the kind of cancer, a minimum of 10-year latency. Some have 20, 30, 40 years latency. So this kind of study, where we had samples and we could go back and look and determine what the exposure was during a critical window, is extremely important. Now, I said I want to kind of go back and forth between some of the mechanistic data and the human data, and here's some data looking at the development of a mouse mammary gland with a chemical, with a chemical called perfluorooctanoic acid, otherwise known as PFOA. This is a member of a family of chemicals that is used extensively for stain resistance, for water repellency, and we're beginning to realize, I should say, that the major manufacturer in the US of this compound had agreed to stop making it as of last year also. But we've had some of this data for quite a while. And the other problem is some of these kinds of chemicals that are much longer chains can actually break down in the environment to give you this. So they may not make this, but we're still getting exposed to it because it's breaking down. And this is a very, very persistent chemical, both in the environment and in our bodies. So this is just some data from a mouse, and this is, this is the control mouse, and you can see that this is just when it's weaned, so this is 3 weeks old for a mouse. And this is when it's a mature adult. And what you can see is that you have a very dense structure. These are the ducts that are being developed. Here you have the ducts here, extremely important for the mouse to be able to lactate. But what happens if you get exposed to increasing levels of, and these are low levels of this chemical, only during gestation? If you expose the mouse once it's born, you have absolutely no effects on the developing mammary gland. But what you find here is look at the difference, compare this structure with totally looks different from this, much less dense branching than, for example, what you have here. And this has consequences which have actually been documented on the ability of that mother to feed her pups. Okay, now there's also data that exposure to these kinds of chemicals are associated with an increased breast cancer in women as well. And again, this is, and I have to say, this is what I just said I hate, because it's looking at the levels of these kinds of chemicals in breast cancer patients and comparing it to levels and controls. But I've also told you that these chemicals hang up in our body for a very long time. They've got half-lives on the order of 4 years or more. So that this is actually an indication of what the exposure was as well 10 years ago or maybe even longer. And what you can see is the chemicals from this class, for example, this is the one I showed you the data from the mouse data, has a significantly increased risk of breast cancer than the controls do. So we see it in the animals, we're seeing it in the people. Some other data related to importance of early life exposure and low doses of chemicals. This is kind of some neat studies. These haven't been published yet from some of my intramural investigators, but these were some studies that they were doing in rats. And what they found, for example, is that they exposed the animals to 2 very, very different levels of arsenic. Now, the World Health Organization recommendation for arsenic in the drinking water, it should be no higher than 10 parts per billion. Well, if you go up to a 4,000-fold higher level of exposure, okay, those 2 doses were compared. So a low dose, which has environmental relevance, I can tell you at least in the United States, there are over 16 million people who are routinely exposed in their drinking water to levels of arsenic that exceed this WHO level, and believe me, in many parts of the world, it's much, much worse than in the US there. But what you find is, here is the control, and what you find is that both the low and the very high dose, we're finding a shift in vaginal opening has to do with when the animals reach puberty. And we also, which we see over here as well, we also find, and there is data from epidemiological studies as well as some other experimental animal data, is that the body fat content is increased with exposure. Again, this is all in utero only to arsenic. And again, very similar at both the incredibly high dose and a very, very environmentally relevant dose. And that we also find not only are they fatter, but they are also developing glucose intolerance, which is on the pathway to a diabetic response. And what you can see is, again, here are the controls, and here are the arsenic-exposed animals. Now, I mentioned before the issue of male reproductive development, and this is some very important data, I think, coming out on phthalates. And I know Andreas just told me today that he has just finished working on a large project with others in the US, as well as from other, on the problem with phthalates. Phthalates, there is a whole family of these chemicals. They were used in baby toys. They're used to make plastic tubing. They're used extensively in cosmetics and personal care products. And some of them, in fact, can block the action of testosterone or act against the male reproductive hormones. And what you see, this is data from mice, and what you can see, AGD is anogenital distance. This is the distance from the base of the penis to basically to where the anus is. And in males, that's supposed to be longer than in females, okay? So if you have a shorter anogenital distance, it means you have demasculinization going on. And what you can see here is that this is one of the phthalates, dibutyl phthalate. You have a decrease here. If you look here, if you're looking at the testis weight or Sertoli cell number, which are the cells that are important in producing, that really lead to and constitute and produce the sperm, what you see here is there's also an association. The shorter the anogenital distance, the smaller the testis and the smaller the number of cells. Well, guess what? That effect is not limited to rats and mice. It's also been documented in humans. So Shanna Swan has done some very, I think, important studies where she has actually looked at the anogenital index. And here she looked at little boys from the ages of 6 months to 3 years. So she had a control for the fact that some of them were a lot bigger than the others. So it's a ratio of the anogenital distance to the body weight. And what you can see here is that as the level or as the concentration of phthalates moderated by a phthalate score increases from very low to very high levels of phthalates, what you see is that you have the anogenital index changes and is indicating of a shorter. So it's, again, an evidence for demasculinization in these little boys. Now, what does that mean? You know, what are the impacts of that? Do you know 20% of people going to assisted reproductive technologies, going to clinics to do in vitro fertilization or have help, are based, at least 20% are because of men who have very low sperm count. And what we're seeing here is that the sperm concentration, and this is a follow-up study in young adults, young adult men, and what you see here is that those with the shorter anogenital distance statistically are at greater risk of having low sperm concentration. And I can tell you this is starting to get in the region where at least a friend of mine who's a reproductive specialist said in his patient population, these men become sterile unless they have some kind of assist. So I think what we've seen in the experimental animal models and what we're seeing in the young boys has a physiological consequence, which is low sperm count. So this is just a quick dioxin slide, and I don't want to go in detail, but the important thing is that we know that in experimental animals, dioxin or TCDD, often called the most hazardous man-made chemical, very involved in Agent Orange, for those of you in the Vietnam War and so on, that has an antiandrogenic effect, and we see that that can impact anogenital distance and penile length as well. And this is from a very recent study that just came out of a large cohort involving children in Crete and children in Greece. So not limited to any one area. Now I mentioned, and I gave you a little bit of data on arsenic leading to, at least in the experimental animal models, being associated with an increase in body weight and then potentially leading to diabetes in the animals. Well, I think, as you all know, obesity is a huge and growing problem. It is not restricted to the developed world, although clearly my country has a huge problem, and my part of the country, the southern part of the United States, has, overall in the United States, 2/3 of our people are overweight, and half of those are actually obese. And in my part of the country, it's much worse. And there are a lot of reasons possibly for that. But you can see, even for example, in some of the least developed parts of the world, the prevalence obesity is still about 10%, and it's increasing. I don't know how many of you travel, but as you travel in areas where you never used to see fat people, you now see a lot. And I'm really concerned, it's not only fat adults, but we're seeing fat little kids. And I think that doesn't bode well for our health, and I know we are concerned that this will be the first time that children, in a long time in many parts of the world, that children don't live as long as their parents, and that may be because of all the adverse effects of obesity. So what we're beginning, we actually held a workshop It's hard to believe it was exactly 3 years ago now, but we looked at the role of environmental chemicals in obesity and type 2 diabetes. And there's been a term coined for these environmental chemicals that play a role in obesity called Obesogens. And what we found is that, for example, nicotine can act as a developmental obesogen, and nicotine, I'll show you some data from tobacco smoke where we believe it's the nicotine that does it. We know that certain, for example, environmental chemicals like BPA, and I'm going to show you some of the data for the effects on the pancreas and the cells there. We know there's a significant association between persistent organics, dioxins, perfluorinated compounds, DDT, and type 2 diabetes. And we're finding that multiple kinds of pesticides may actually increase diabetes and obesity. So I'm never going to say you can't exercise, you don't have to exercise, and you can eat as much as you want. But are we putting people at increased risk? Are we making it harder for people, because their metabolic programming has been shifted to push them in the direction of obesity? So this is just some data on tobacco smoke in utero, and there have been a huge number of studies now. In fact, we've looked at over 24 different studies based upon the mom. If the mom smoked during pregnancy, by the age of 10, her children have an increased risk of obesity and type 2 being overweight, being obese, and type 2 diabetic. And what we're also finding is that there appears to be an increased risk, again, depending on the exposure of, for example, here's the obesity. And while these in this study were not statistically significant for elevated blood pressure and type 2 diabetes and gestational diabetes, there are trends there that are indicating that this is true in this population as well. Whoops. So I've mentioned BPA a couple times. I don't know if BPA is as hot button a topic in the UK as it is in the US, but it's huge and in the US. And my organization had issued a report about 5 years ago, it's more than that now, 2008, where we said we had some concern for the neurodevelopmental effects, meaning it could affect brain development, and also for the effects on the prostate gland. Our Food and Drug Administration has supported that conclusion, and for example, BPA has been removed from baby bottles and removed from baby sippy cups, but it has not yet been removed from the linings of cans. It has not yet been removed from its use in dental sealants or in thermal paper, and there's growing concern. And I could say the marketplace is acting, but one point I'll just allude to here is the marketplace acts. So some of the manufacturers of the chemicals say, okay, we won't use them anymore, we'll use something else. But at least in our country, there's no requirement to test anything for safety before it goes on the market. And some of the substitutes may be worse than the compound that we remove. So, but anyway, this is some data on BPA, and this was an analysis of 8 different human studies looking at BPA's association, and we see in all cases there was an increased risk for diabetes, for glucose homeostasis, and for overweight and obesity. So, and again, there have been several studies since then which support this potential association. Again, way too complicated a slide, I apologize. But this is new data that came out of one of our grantees, Leo Trasandi from New York University, and he basically was mining data. We have a biannual survey that's being done in the US called the National Health and Nutrition Survey, survey, whatever. And they measured about 250 different chemicals in approximately 8,000 to 10,000 people every year, and they take extensive health data on these people. It's a cross-sectional study, so you don't know what the person was exposed to before, and you don't know what happens to them later. But it's still a very rich source of information, especially to generate hypotheses to test, but also sometimes we get understandings from this if we have, the animal and the mechanistic data to support it. And what this showed was that in children, those with higher BPA were more likely to be obese, overweight and obese. Now, it doesn't address the question, is there something about being obese and overweight means that you're, say, eating more foods with BPA in them? You know, maybe more canned foods instead of fresh fruits and vegetables? We don't have the answer to that yet. But again, mechanistic data. And here's some data showing from cells in culture, and these are mouse cells in culture, but what you can see is, I should point out, this is what happens with the natural human estrogen, estradiol, and what you can see is that as you put BPA in the culture, you get more insulin release from the pancreatic cells. And what you also see is when you get high doses of BPA, that doesn't happen. This occurs in a very low dose BPA range. And again, the same kind of thing from in vivo studies, that this is what happens if you just use control cells, and if you add BPA, actually in vivo to the animal, you get an increase in the insulin release. So I just had to mention air pollution because, as I mentioned, air pollution is now declared a known human carcinogen. But air pollution is associated with a whole adversity of health effects. And there are a couple reasons that that could be. Air pollution, one air pollution is not the same as another. You know, you have ozone, you have sulfur dioxides and nitrous oxides, you have carbon monoxides, you've got all kinds of particulate matter, which contains, for example, polycyclic aromatic hydrocarbons, which are developmental toxicants, immunotoxicants, and carcinogens, for example. But there's a host of adverse effects that are now being reported from air pollution. And I think an important thing is we're also beginning to see these associations with developmental disabilities, especially neurodevelopmental disabilities in children. And this is some data just showing that, for example, that the increases, for example, in attention deficit hyperactivity disorder and the autism spectrum disorders has increased dramatically in the period, for example, 10-year period. And I can tell you that at least with autism, that increase appears to be continuing. We're expecting results from the Center for Disease Control and Prevention next month. And what I'm hearing is that instead of the estimates being 1 in 88 children at the age of 7 to 9 years of age will be somewhere on the autism spectrum, that we're expecting it to be considerably more than that. And I can tell you that the data from Korea suggests that it's 1 in 37 children, and a study that we just funded that was done in Somali children in Minneapolis was that 1 in 32 children were somewhere on the autism spectrum. So we're seeing, and again, we're seeing big increases in a lot of these neurodevelopmental issues. So what could be causing this? Well, I think we all know that lead is a neurodevelopmental toxicant. We all know methylmercury is. There's lots of data with PCBs doing it. This is some data with pesticide exposure. I don't know, again, whether chlorpyrifos, which was an organophosphate pesticide, whether it's banned in the UK. It was banned in residential use in the US in 2002, but that doesn't mean that some people still aren't using it. And if you happen to live in an agricultural area, it's still going to be used in the farms. So children living in those areas are being exposed. And this is a study where they did MRI imaging on the children looking actually at brain structure. And what you can see is these are data showing that as the chlorpyrifos levels increased in the brain, there are changes that are going on actually in brain structure in a number of different regions of the brain. So these were children who were born when they were born between 1998 and 2000, and then they were looked at 10 years later. So most of their exposure occurred, again, in utero, because actually this chlorpyrifos level is not the child's level. This was what was in the mother during pregnancy. So again, the window of susceptibility issue, the key impact of early exposure, permanent changes on the structure of the brain. And we have data from at least 3 different cohorts now of children in the US who have been followed since before they were born. Now they're at the ages of 13 and 14, showing that elevated chlorpyrifos exposure in utero is associated with decreased IQ in the children. Now, the IQ deficits, we're not talking about a 20 or 30 point deficit. We're talking like lead, 4 points, 5 points, maybe 6 points deficit. So I can't look at you, I can't look at Susan and say, darn Susan, if you hadn't had all that prenatal or early life lead, you'd have 5 more IQ points. You might have. But you can't look at that in an individual. You have to look at a population and see the shift. And we're seeing that, for example, with these OP pesticides. So what do we need to do? Well, at least we estimate in the US that there are at least 80,000 chemicals in commerce, and less than 20% of them, or about 20% of them, have any degree of testing. And we're really concerned about a fair number of them. In the EU, I think, under the REACH program, they're estimating 143,000 chemicals have to be looked at. But again, in the US, we have no requirement to look at them before they're put on the market. And most have little data at all. And we're using these biomonitoring programs to identify what we can find in people, and maybe if we're finding it, maybe we should try to understand whether it's a problem. So how are we going to begin to test these? Well, I think a really important thing is that we can't do loads of human studies a year, and we never do controlled human studies. We're never going to intentionally expose people to a chemical for which we have concern. So our human studies are epidemiological, they're observational, and there can be issues in interpreting them. We can do our standard rat or mouse kind of studies, and in the whole world, there may be only 10 to 100 at most being done a year that are looking, for example, at long-term effects, especially going to a 2-year study, for example, to look at cancer. Well, we can use some, for example, alternative animal models. Animals that, for example, are not mammalian models. We can use zebrafish, for example, which are one of my favorite models, because you can actually, they're transparent when they're developing, and you can actually look in them and see the heart start to beat. You can see the brain form. They're really incredible. And their whole life cycle is only 3 months. Pretty nice, as opposed to rats where you might have to wait a couple years. Or you might use C. elegans, which are those little round worms, or we might use Drosophila, the old genetic favorite. We can start using in vitro or cell-based assays, and we can look at critical pathways, and we can start doing things computationally, but somehow we've got to put this all together, and we've got to increase our throughput. Because if we can only test, say, 10 to 100 chemicals a year, we're never going to get to that 80,000 chemicals that we have to know something about. So about 6 years ago, NIEHS, and the National Toxicology Program, got together with EPA and the director, he was the head of the, now he's the director of NIH. At the time, he was director of my sister institute, the National Human Genome Research Institute, and they proposed a program called Tox21, or Toxicology in the 21st Century. And this is a program in which we're trying to use high throughput screening approaches, where we're looking at different kinds of endpoints, and we're looking at hundreds of different kinds of endpoints, and we're not looking at 10 or 20 or 100 chemicals at a time. In fact, we have now looked at over 12,000 different substances in hundreds of different kinds of biochemical response in vitro assays. And I can tell you, while I was very skeptical of this, because I tend to have done mainly whole animal studies or even human studies, when you look at this many endpoints for this many chemicals, patterns begin to emerge. And you can identify which chemicals are the bad actors versus those which appear to have very little effect in a number of different systems. We're also, and I should say that the picture up here is the robot that we use, which can run through 10,000 chemicals through 70 different assays easily in a year. We're also looking at something called the Thousands Genomes Project, in which we have cells that we've isolated, almost 11,000 different lymphoblastoid cells derived from the lymphatic system from 9 different ethnic groups from 5 different continents. And we looked at, for example, just simple assays, well, they're not so simple, but for cell viability. And we looked at almost 200 different chemicals, and what we see is that there is over a 200-fold variation in the response of these 1,000 different cell lines. So that's very important, because in standard risk assessment, people say, oh, there's only a 10-fold difference to account for all the individual differences. This is just based really on genetic differences between these cells. This isn't accounting for life stage differences or anything, over 200-fold difference. And then we have another approach. We're saying standard animal testing usually uses inbred strains of rodents because it's easier then not to have a lot of variability that makes it hard to look at what you're looking at. But in fact, people in general aren't inbred. So there's this new approach where what's been developed is the diversity outbred mouse and the collaborative cross, which spans the variability in mousedom. And we've done some studies with a number of different chemicals. For example, benzene, which you've probably heard of, and we know it causes leukemia in people, as well as a number of other adverse health effects, neurodevelopmental as well. And what you can see is that when you look at this variability in mousedom, there are some mice who don't respond at all to benzene and others who are extremely sensitive. And we have, again, a variability of well over 200-fold. So we need to begin to understand, you know, how variable the population may be for, and how different the susceptibility may be. And we need to be careful in interpretation of some of our animal data, especially if we find a negative response and assuming that that means no human will respond. Because maybe we just used the wrong animal model that was not sensitive to what we were looking at. So we also are looking at the identification and validating of new alternative test methods. And the biggest thing I'd say we're taking a huge stab into, and I know you are here as well, we've got a whole effort called BD2K, which is big data to knowledge. There's huge amounts of information being generated, not only from all the genomic data, not only from all the exposomic data or all the exposure data, but everywhere. And we need to begin to understand how we can utilize all this data. So if you know of anyone who's interested, a student who's interested in a career, if they've got some mathematical capability, tell them I've got a job for them in bioinformatics. So in closing, I've talked somewhat about the environment. I've made the point that prevention is really the key. I think we've got to stop talking about is some health condition genetic or is it environmental? Because the answer is it's always going to be both. It's always going to be an interaction between the two. Some are maybe more genetic, some may be more environmental. I've stressed the point that when exposure occurs can be absolutely critical. We have different windows of susceptibility where if you get exposed in utero, you can have effects that last for a lifetime. There are other sensitive windows. The infantile period, puberty is a really susceptible time because the body is rapidly changing. Pregnancy for a woman is a very susceptible time because her body's rapidly changing. And then clearly in the elderly, there's differential susceptibility as well. We need to understand what the hazards are to this plethora of compounds to which we are exposed, but we need to think about exposure at levels that have some meaning, environmentally relevant kind of levels. We can't do anything about our genes. We can't change them, but we can change our environment. And this is an opportunity for us to go forward to improve our health. So I'd say the take-home message is that in the same way as medicines produce effects in the endocrine system and other systems, environmental chemicals can produce unwanted endocrine effects, resulting in an increase in human diseases. These can have significant population-level impacts, and therefore, instead of telling someone, you can't do this or be careful of what you eat, we really need public health measures to help prevent disease. So thank you.
Thank you, Linda, for a wonderful lecture, very interesting and very captivating. We've got some floating mic. We've got somebody standing there with a mic somewhere. Have we got questions for Linda? Vice-Chancellor.
Thank you very much, Linda. That was absolutely fascinating lecture. I think there's a lot of evidence that nutritional factors, like, for example, protein, lipid in the diet, stress, whether it's emotional or immunological, can produce many different effects, particularly predisposing to diabetes, type 2 diabetes, and also neurodevelopmental effects. And I wonder if there are any studies which have actually looked at the interaction between those lifestyle effects and the environmental chemicals which you're talking about, and also whether in your studies you've got any evidence of gender-specific effects, particularly on neurodevelopmental changes.
So I'll take the second one first. The first is gender-specific effects related to neurodevelopment, and the other is the interaction between, say, nutrition or psychosocial effects. And the answer is yes to both. So as far as gender-specific effects, especially with neurodevelopment, we're finding that with a number of especially environmental chemicals of concern like BPA, we are seeing effects, for example, in toddlers. There have been neurobehavioral effects which have been documented in toddlers in a couple of different studies that we see in the girls and we don't see in the boys. We're seeing effects from a number of compounds. I think it's phthalates, and not PAHs, but I may have that because they both are associated with an increase in ADHD, and they are primarily in the boys and not the girls, which might not be a surprise since the incidence of ADHD is much higher in boys than it is in girls. As far as the issue of either psychosocial factors or nutritional factors and environmental, there's growing evidence for a number of those interactions, and I'm actually going to give you a couple of animal examples first. If you stress, which a lot of psychosocial involves stress. If you stress animals prenatally, for example, not only will their offspring end up being poor mothers, but their granddaughters end up being poor mothers. Now you can modify some of that with certain exposures and certain behavior. But with diet, I actually did a study with one of my students about 5 or 6 years ago where dioxin has been one of my favorite chemicals, so I have to mention it. But we did a study where we were looking at breast development, which we know can be severely impacted by dioxin. And if we put animals on a low-fat diet and expose the pregnant mouse in the middle of gestation to dioxin, the pups were perfectly fine, nothing at all. If they were on a high-fat diet, their breast development was totally messed up. So we're seeing this in a number of different examples, and there is some data coming out from the epidemiology literature. In fact, there's some really new data coming out on air pollution and psychosocial stress, and showing that the health impacts are much worse, again, under stress. Maybe not surprising.
Thank you.
Thank you. Any more questions for Linda? I'm sure there are some.
What do you say to the naysayers who say these chemicals are present in the environment only in very small quantities and they only have the activity several orders of magnitude lower than the natural hormones? How can they possibly be having the effects that you're describing?
Because that's a very good question, because BPA is one of the poster childs for that discussion. And part of that is people are focusing on the classical estrogen receptor binding affinity for BPA to the estrogen receptors, both of the two, ER alpha and beta. They're forgetting that there are other kinds of estrogen receptors now. We know the membrane and the cytosolic. They're forgetting that the estrogen receptor is a pretty complex molecule, and if all you're looking is binding to the classic ligand binding domain, where estradiol binds, they're forgetting about the part of the protein, which is where anti-estrogens can bind, and that BPA has the same affinity there as some of the most potent pharmaceutical anti-estrogens. And they're also forgetting that BPA doesn't just affect the estrogen system, but can also affect androgen, thyroid, and the system that's involved, we believe involved in diabetes and fatty acid metabolism. So people are looking with a very narrow, I would say, blinders on. And I think the other the point is that we're now beginning to generate data, not only in experimental, like mechanistic cell-based studies, but there are animal studies where we are seeing effects at levels of exposure that are starting to be at least at the higher end of the human population. And that's another point. people will say, oh, the average level in the human population is, let's say, 1. But we've got a huge variability, not only in response that I talked about with the cells, or I talked about in some of the others, but in exposure. So there are some people, in fact, when we do these measurements of, say, BPA levels in people, that are 100 times higher than the average. May not be many of them, but they're in the general population. We have no idea why they're exposed. So I think that it's an issue of what's the question you're asking, will help determine where you come down on these issues.
I think Andrew had a question.
So I'm speaking as an immunologist, and one of the issues that we have in immunology is desensitization of receptor systems, which means that the immune system is very good at spotting differences and changes rather than steady states. It's obviously designed to do that. But it makes me ask, to what extent is the kinetics of drug exposure or environmental exposure going to be important? In some drugs, obviously, it'll probably be relatively unimportant, but is there a role for changing levels actually being more detrimental than perhaps constant levels?
The answer is yes. I think there is a role for some of that. We know, for example, that a lot of endocrine systems in the body are not relatively constant, but are actually pulsatile. In other words, what makes the difference between a male and a female in part is whether your GnRH, the stimulating hormone, is pulsatile or constant coming out of the brain. But I think this whole issue of desensitization and negative feedback is very important in endocrinology. And what we're beginning to see is some of these low-dose effects. And I showed you one piece of data there with BPA where we had an effect at the low dose, but we didn't have it at the high dose. And part of that, again, is because you can get receptor feedback and actually desensitization or tolerance develop. And I didn't use any examples of the immune system, but I could have. So.
More questions for Linda? There's a lot of students and postdocs over there in the corner from the Institute for the Environment, I might add. Any questions for Linda? Well done, Chris.
Yay.
So if prevention is going to be better than cure, how do we persuade politicians that that's going to be the case?
We're not in the US, so I hope nobody's going to tell my local congressman. Actually, my local congressman's a good guy. I think one of the things that my institute is doing, and I didn't talk about my institute per se, but we are one of the institutes of the National Institute of Health. We're the only one that's not located near Washington, D.C. We're in North Carolina, the southern part of heaven. What's at least speaking now is money and the dollar. So in our new strategic plan, we actually call that out as one of our key goals, that we have to get economization done of some of the environmental costs and the environmental impacts on health and disease in order to make the case that if we want to save money on health care, the only way we're going to be able to do it is by preventing disease. So a very interesting article was published by one of our grantees, David Bellinger from Harvard, last year, and there have been some other articles I've done it, where they'd actually estimated or predicted the cost of neurodevelopmental effects from only 3 different kinds of well-known neurodevelopmental exposures: lead, methylmercury, and chlorpyrifos, which is an example I gave you. And the total cost of the decreased IQ that is associated, and it's like a 5-point shift, 7-point shift that might occur overall in the population. What that means is as much as you get from all the the genetic birth defects, the medical, for example, children with congenital heart disease often need surgery and may have elevated incidence of IQ deficits and so on and associated, is as much or more than what occurs from all all the different kinds of medical things. And the lead and the chlorpyrifos, and those are preventable. They don't have to happen. But a 5-point IQ shift, while Susan's okay, you know, having an IQ of 135 instead of 140, but you know, you know, but the point is you not only lose people at the high end, much more importantly, you shift the population so there are more children in need of special services. And those children grow up to be adults in need of special services. And the costs just are huge. If we look at the costs of care of people with type 2 diabetes, for example, the cost of heart disease, the cost of cancer, they're huge. And if we can prevent some of these health impacts from occurring and get the economic estimates of what that will, I think that will sell.
Thank you. Question over there?
Microphone. As an economist, I've been sort of thinking a little bit about the issue of how you value and put benefits to the management of hazardous chemicals. And I suppose one of the problems I've got, certainly dealing with my colleagues, I work for the Environment Agency, is that when I ask them, well, what are the endpoints? What are the consequences of actually removing these various chemicals from the environment? They generally don't know. Now, I think there may well be problems with the regulatory model we have at the moment, which is to do with individual chemicals rather than collective groups of chemicals. But I suppose the problem we've got at the moment, it seems to me, is that by the time we have the information which is actually needed to do that benefits assessment, in fact, it's really rather too late because the damage is already done. You've mentioned you know, probably the best source is actually epidemiological studies. So I suppose the question in my mind really is how do we anticipate problems in a sense and actually be able to identify problematic chemicals before that evidence actually accumulates and allows us to actually be properly precautionary in terms of the way that we manage chemicals.
Yeah, so I'm not sure I have a real good answer for that. I think part of the issue is I've talked about these new approaches to doing testing where we may be able to identify bad actors, and under the REACH program, the bad actors hopefully would never even get on the marketplace. There is a movement in our country to start to possibly change our legislation to require some testing before chemicals goes on the marketplace. I don't think it's not going to happen this year. Maybe next year or the year after, if we're lucky. But I think that if we can identify where the compounds that we have concern, as we begin to develop patterns, you know, a kind of do pathway analysis, kinds of understanding where the problems are occurring, I think we can at least begin to avoid things that may be a problem. When we talk about, though, there have been some economizations done related to certain complex mixtures like air pollution. And in the United States, we estimate that before we lowered the regulations for particulate matter in the air, that we were having at least about 167,000 deaths associated each year because of air pollution. And you know, if people die, then you can calculate the years lost of earning power and all that kind of thing. And those kinds of estimates are being done. But I totally agree. How do we develop a prospective look instead of waiting till the problem happens?
There was a question over here.
You mentioned the role of environmental chemicals in things like obesity and reduced semen quality. And I was just wondering, is it possible to sort of attribute the significance of the role of environmental chemicals in those situations? Because obviously there's lots of factors, including lifestyle factors and how much you eat and things like that. So is there a way to determine what the significance is?
Yeah, so I think in the epidemiological studies, people are doing the best that they can in epidemiology, which are observational studies, to control for all the other factors that you mentioned, lifestyle factors, nutrition, socioeconomic factors, mother's education, You know, how much TV does the kid watch each day? All those kinds of things. And you still come up with significant role for the environmental chemicals. And again, we've got to have this two-way street between the epidemiologists and the basic scientists and the toxicologists, like maybe it's a three-way street, you know, because to understand, sometimes the epidemiology has a finding that we had no idea that it was happening in the basic science of the tox. Then we have to go back and see, can we find, does it occur? Very often we find something in the toxicology literature or in the basic mechanistic literature which says, uh-oh, look what we're finding. Might this happen in the human population? And then we go looking. So some of the stuff, for example, I've talked about IQ deficits in children, but for example, the flame retardants. There are now several studies showing that prenatal exposure to the flame retardants is associated with lower IQ in kids. We've got, again, mechanistic and animal data that support that finding. So I happen to be a big believer in biological plausibility. I also tend to believe that nature is pretty conservative, and when it finds something that works, it tends to use it repeatedly, maybe with some perturbations or permutations, but it tends to use it again. And I also believe that while animals are not people, people are animals. So.
Any more questions for Linda? We've got quite a few questions there. That's quite good.
Great.
So what I'm going to do now then is just turn to Andreas to give the vote of thanks.
Oh.
Come sit down.
Oh, okay, thank you.
So Linda, I'd like to thank you very much on behalf of everyone here and the university for coming along such a long way to give this very stimulating presentation to us. The messages you make are really timely, and I've always admired you for being able to put across these points so eloquently. We're so used to hearing, in many cases, take for example breast cancer, the rates are going up, as we all know. We're so used to hearing, yeah, either you can't do anything about it or it's the women's fault because they smoke or stuff like that. But the point that is so important for creating a prevention framework for these diseases, not only breast cancer but the others, is so important. And I'd like to thank you very much for bringing this message across to us tonight. So thank you very much.